Yes, I have it. Thank you very much. So I'm going to speak to you about exosomes, which is a new. It is the last years a good platform to not only make a diagnosis and understand what happened in the brain. This is an example I bring from the brain, but it can be used on different issues and different diseases. One is the diagnosis. The second is the exosomes are small vesicles, we're going to see, that they can also be loaded with drugs and used for treatment. So let's try to see this job. And we are interested, my lab is working basically on 3 research lines, which is trying to connect how psychological day life stress, that researchers know a lot, has to do with depression and Alzheimer's disease. Why depression and Alzheimer's disease, which they seem irrelevant? Does anybody knows if there is any connection between the 2 diseases or anything? No. So you think there are 2 irrelevant things, correct? 2 irrelevant diseases. So the answer is the stress is connecting them, because chronic stress, psychological stress, is a risk factor for depression, but also, based on the work we have been developing in the lab for the last 23 years, chronic stress is also a risk factor for Alzheimer's disease. So if you're more stressed, so please don't stress your students behind around. If you're stressed, you can also have high risk to develop Alzheimer's. And imagine that you're stressed, so you develop depression, and then you have high risk. If you have depression, you have high risk to have Alzheimer's. So what we say in the last 23 years, starting from the work I developed in Max Planck Institute of Psychiatry in Munich, that chronic stress is probably the link between the 2 diseases through common neurobiological mechanisms, which we try to understand. Then we try to use some therapeutic approaches, because there's no, as you know, drug for Alzheimer's. And also, while we'll have a lot of antidepressants, drugs, we know that something between 30 and 40% of depressed patients do not respond to the current antidepressant drugs. So we're taking different drugs, like based on antisensory glucotides, which, you know, they are small molecules binding on mRNA and destroying the mRNA of the pathological protein. In that way, you reduce the pathological protein. Or also we use psychedelics or cannabidiol, try to treat with different ways depression, Alzheimer's. And also we try to understand whether all these treatments or pathologies we can have information through exosomes. So, okay, no. Okay.
I will be here behind you. Continue, my friend Jonas.
Okay, that was the past. So basically, in the next slide, whenever it appears, the last slide, this one, I'm going to just press one more, or I can press.
You
more.
can press, yes.
So this is the main work we developed. So we believe that chronic stress through the main stress hormones, glucocorticoids, are activating the 2 main pathways of Alzheimer's disease, amyloid beta and tau. Amyloid beta is overproduced in Alzheimer's disease brain, and tau is hyperphosphorylated and aggregated abnormally inside the brain. These are showing all these different levels of damaging the brain. And then we have different ways try to block all these pathways and to provide new therapies. So I'm going to skip this. You know that we have a big increase of Alzheimer's. This is supposed to be a number 145%. So based on the progress in biomedical sciences, we have a lot of treatment of diseases that we never thought. For example, HIV. You see, it's reduced. It's a lot of reduction of the deaths we have from HIV. However, diseases like Alzheimer's disease, it's very much increased, 145. The number is supposed to be 145. So this indeed create a big rush for that. Just to tell you basic things for Alzheimer's, for people that you don't know. It is a neurodegenerative disorder. It is basically on memory and moods and personality problems, but this is the main problem. What do you see here? Atrophy, exactly. So we have even macroscopically, or the neuronal level, or even in the synapse, the way that the neurons communicate and store new memories. They are reduced. This is why Alzheimer's disease patients, for example, they don't remember because they lost the ability to form new memories, to create new memories. For example, now, if you want to remember my difficult name, which is Ioannis or Yanis or Anis, based on Tunisian name. So it's Ioannis. So you're easy to memorize. You need to make new synapses. You need to make new memories. And now you can. But Alzheimer's disease people do not have this ability. So, and the second thing that we usually check is what I told you, the amyloid beta, which is a small peptide aggregated coming from a big membrane that we cut separately, and it gives this amyloid beta here. And then we have these tangles. This is all histological analysis of the brain of Alzheimer's disease, which comes from a tau protein. Tau protein is a cytoskeletal protein that binds to the cytoskeleton and tries to stabilize it. What happens in Alzheimer's disease is that tau is hyperphosphorylated, so it's damaging its function. Now, in this slide, we have seen a lot of targets for clinical trials. These are 51 agents in clinical phase 1 against tau, against amyloid beta, against neuroinflammation. And there's also in clinical phase 3. However, look at this, big failure. Why? The question is why? Do we think, for example, that what we believe for Alzheimer's disease, the way we see the disease is wrong? Is it possible that we have more than one type of Alzheimer's disease? And I'm not talking about genetic familial Alzheimer's disease or non-genetic type, which is the sporadic. I'm talking about if we see the disease as one disease. And this is where the precision medicine, the new era of medicine comes, trying to provide a more holistic means the whole, understanding. So I told you that there are some molecular changes here where you can see increase that there leads to increase of amyloid beta and increase of pathological forms of tau in the brain. That's okay so far. And these are responsible for the synaptic dysregulation in different brain areas. And at the end, we have the memory problems and the mood problems in the Alzheimer's patients. However, there is a period between the first molecular lesions in the brain until the appearance of the first symptoms, which will drive me to the doctor and say something is wrong with my brain. I park my car and I cannot remember where it is the whole afternoon. There's a problem, So this period until the first molecular change in the brain and the first symptoms can be as long as 15 or 20 years. Wow, that's a long period, isn't it? So, in other words, I can probably have started Alzheimer's disease already in my brain, and I will know in 15 years. And this is where precision medicines take into consideration parameters that are different on each of us, like physical activity. We produce neurotrophins that help synapses, that help neurogenesis, Mediterranean diet. I hope not many of us eat burgers and McDonald's. Okay, mental engagement. What the Americans say, use your brain or lose it. Use it or lose it. And there are all these parameters, they try to slow down this process, this damaging process. Where there are also, on the other hand, risk factors, precipitating factors, such as chronic stress, the work we are developing, such as cardiovascular problems or inflammation in the brain, that accelerate the whole fast. It makes them go faster. So, yes. So, what the precision medicine suggests is that we have to move far from the old way of thinking in medicine, which we call it one size fits all medicine. What does it mean? This means that probably we have more than one subtypes of Alzheimer's disease. So, which you can see with different colors. What I'm saying, some patients, the first symptoms of some patients is memory problems, as I told you. But some are having motor problems, like don't gain well. Some, they have a lot of amyloid beta and less tau in their brain. Some, they have a lot of inflammation, but some don't. So what the precision medicine is trying to understand is, if I go to the doctor, they will try to, first of all, do a stratified medicine approach. So create these different subtypes and characterize well. How we do that? We try to based on taking into consideration parameters such as lifestyle. Do I work with a lot of stress? Do I work overnight and switch my sleeping rhythm? Do I live in a cities with air pollution? Do I have cardiovascular problems and history? But also taking my omic analysis. So we take blood and we do analysis on our blood. And trust me, in a few years, it will be cheap for everybody to have some, take blood and have proteomic analysis, epigenomic analysis, metabolomic analysis based on our blood and components of the blood. Not only the cells, but also components like exosomes, which we're going to say. So the idea is that I go to the doctor, and the doctor compares all this data of mine with what we call big data. So we're talking about millions of patients before me, and compare my data to this big data cloud. They are going to say, oh, John, you belong to that category, so I'm going to give you that drug that is going to help because you have a lot of inflammation, so I will start treating you with inflammation. Or, John, you belong to the other one that has a lot of tau, so I'm going to give you another drug. So this is what we previously called it personalized medicine. This term is not anymore correct because I'm not going to give you a personal drug for you and another for you. That's why we speak about precision medicine now, okay? But to do this, to classify me in the category, they need better biomarkers for me. So if I have better biomarkers, for example, this is the decline we usually have by aging. For us, that we cross the 40, 45 years old, working memory is going down. You can see this here, but this is healthy. Now, what happened in Alzheimer's disease? We have a cognition and functional decline very sharply. So, and this is the prodromal phase of Alzheimer's. We call mild cognitive impairment. We can have or not have something. So if I have better biomarkers, I will be able to understand as early as possible, or even before, what happens in my brain. Then, if I understand what happens before, I can see, okay, that I have this stress, I have this risk factor, I need to remove it. And this is what we call prevention, primary prevention interventions. Remove risk factors from our life, but I know that something is wrong. So I go and slow down the things that are accelerating my brain pathology. And for sure, if I have better biomarkers, I also will see if a new drug or something works at the molecular level in my brain, if it reverses what we think it reverses. So here is where the exosomes are coming. So what are exosomes? Is these small vesicles with double membrane that they secreted from all the cells. The main hypothesis of exosomes coming from cancer, do we have people working with cancer or coming from cancer field here? A lot of them. So, as you know, cancer, in compared to brain science or neuroscience, they're much more advanced. So they start speaking about exosomes many years ago and saying that exosomes secreted by a carcinogenic cell is secreted to the place where there will be the metastasis, So the exosomes go there, transfer material like proteins, RNA, and they prepare the cellular milieu, the base, the place, for the metastatic cell to come and make a new metastasis place. So something similar, we believe, it happens with brain pathology in neurodegenerative disorders such as Alzheimer's, Parkinson. So this is, for example, a disease neuron in the Alzheimer's disease brain, full of tau and hyperphosphorylated tau, damaged tau. So this neuron, because it's full of tau, it secretes exosomes that exactly they transfer what this neuron has. What this neuron has, a lot of tau, a lot of this, a lot of the other, because they have disease. So exosomes can transfer protein, microRNA, DNA to the next cell, which is synaptically connected. And then this healthy neuron starts to have pathology too. So first role for exosomes is to transfer pathology between different brain areas, for example. Second role, what are you doing? Just tell me what are you doing? Okay.
Just use the-
Okay. So how these organelles are produced, it is part of the endolysosomal pathway, which is a pathway that usually leads old, used, damaged proteins to the lysosome for degradation. And an alternative way before the multivesicular bodies join the lysosome is to be secreted outside the cell in forms of exosomes. Okay? Yes. So the whole idea is that I move from the brain pathology to the brain images that indeed mirror what happened in the brain, because the exosomes transfer information from the cell of origin. And then I can take them and collect it in the peripheral blood. Because, as you see in this scheme here, they can cross the blood-brain barrier and we can collect in the peripheral blood. And what is better than having such an easy biofluid than going and take CSF? But it is very difficult to have it. There are ethical issues in some countries. They can be dangerous, blah, blah. So blood, we can have it everywhere. For sure, all cells, here you can see, for example, that neurons, we call also EVs, extracellular vesicles, because exosomes is one of the category of the extracellular vesicles. And it's not so easy to distinguish from other. So we prefer to say in science, when we write papers, small extracellular vesicles. Because yes, it is exosomes, but we cannot really distinguish so far exosomes from other microvesicles, but we are in this correct range. So again, the idea is that brain pathology, it secretes the exosomes that they transfer information from the pathology, and then I can collect in the peripheral blood for reliable diagnosis and prognosis. Now, so far, and you're very well, in this at least course, you're very well familiar with papain and digestion. In order to collect all the previous methods, in order to collect the exosomes from the brain tissue or any kind of tissue, they use mechanic and enzymatic separation of the cells in order to take the EVs that they are between the cells, because they're secreted, correct? However, this creates a lot of problems. I don't want to go to many details. For example, sometimes if you break the membrane, this is the types of extracellular vesicles that I was referring. So you see there's microvesicles that they are produced by bumbling of the membrane. This is the exosomes. This is the exosomes through the early endosomal multivesicular body, and then they go out. And there's also apoptotic bodies, it's much, much bigger. So when we use this classical papain digestion and mechanical digestion separation, we break some membranes, we do some artificial things that supposed not to be exosomes. And because I cannot distinguish exosomes which are out from inside, then we use another technique, which we published, and in collaboration with the cancer guy, Bruno Costa and Dimitris Kapoyanis in Johns Hopkins and Clarissa, I and Clarissa we are very privileged to have a great MD-PhD student that now is working with Dimitris, postdoc, he's a psychiatrist. We develop a new method when we allow the tissue to release itself spontaneously, the exosomes. So we avoid a lot of problems. And this is not only for brain, it works in human and in mice brain, but also it's good for other tissues to allow the cells or the whole part of the tissue overnight to release the exosomes that they want. Okay, so this is the process. I don't want to go. It's basically ultracentrifugation. You don't need huge amount of money to do this. It's just easy. But I want you to show ways that we can detect and monitor exosomes and be sure that these are exosomes. So the main way, by at least you do it once, to know that these are exosomes, to see them, is the cryo-EM or EM. And you can see these nice photos we have here. And then you can also, in the photos of the electron microscopy, you can also measure the diameter. And as you can see in our method, which is the red, you can see that our method, it produces much more clear and much more many exosomes than the old traditional method. Why I say that? Because, for example, this is another machine that we use to measure the size and the number of the particles, exosomes. You see, for example, that the red, this is a human tissue, this is mouse tissue. We have double the amount, almost double the amount of the vesicles that are in the size between 50 and 150 nanometer, which is exactly the size of exosomes. Where bigger apoptotic bodies or others are reduced. So our technique seems to give a more pure amount of exosomes than the previous technique. And we confirm also by proteomic analysis in this first category that you have in, I don't know if you can see from the back, is double the amount of proteins related to extracellular proteins, suggesting again that this method gives us a purer yield on exosomes. And we also are able, by using a different technique, something happens with my voice. Sorry. A different technique where we can divide it different categories. I don't want to go to details because that's not the purpose, but we are able to isolate different subpopulations of exosomes based on the membranes, the transplants they have in their surface. And this is indicative of some cancer type or some that they transfer mRNA than others. And also there is a new technique, ExoView, which is interferon-based label-free measurement of what is inside. Probably you cannot see here, but these red dots is what we have inside. So we are able to measure tau, for example, inside these exosomes. Okay. And for you, that's also important, working with cells and organoids and primary neurons, that you can modulate or block or increase the production of exosomes. And you can activate, for example, neurons or tissue in order to produce more. So neuronal activation, for example, is well known to trigger the secretion of exosomes. And there's also drugs we can block with the synthesis, the general synthesis and secretion of the exosomes. And also you can label them. There's a lot of dyes. We use DIL, so it stains the membrane. It's a lipid incorporation staining, and we can follow. I don't know how you can see from there, but we inject from one area in the outer molecular layer of the hippocampus, and the exosomes were picked by the dendrites of the cells, moved to the soma and moved down to the axon. And for example, we inject in the outer molecular layer here, you can see the red, but nothing in the first day, nothing in the granular cell layer here. And then 4 weeks later, we check that, yes, we have still the signal there, but now we appear to have, sorry, to have more signal of the exosomes in the granular cell layer. For sure, it's not the best label, and we're expecting more techniques to label it for free. But it is something that you can use now. And there's a lot of improvement the recent years of new tools to use them. And so I told you that, okay, in the brain or different tissues, you see this movement of exosomes as a communication between cells for good or bad, because exosomes also help, communication between cells, help transmission in the brain. But also when the cells are diseased, they send disease messages to their next. But also we said we can take them in the blood vessel. Yes, but I told you that exosomes are secreted by all our cells, by ovaries, by cancer, by heart, by column. Every cell secretes exosomes. So in my peripheral blood, I'm going to have a huge pool of exosomes from everything. But I want to know what happens in the brain. And as you know very well, in biological issues, the main issue is to reduce the noise that really influence what you are trying to measure. So what we have done, we are using techniques that we try to separate, isolate exosomes based on their cellular origin. For example, I want to separate exosomes coming from the neurons of my brain from the rest of the pool that I have everything inside my blood, plasma, for example. So what do we use? We have, and this is a big challenge for the field of exosomes, try to identify proteins in the surface, the membrane surface of the exosomes, which are unique for each cell type. For example, L1CAM, ATP5180. It seems to be neuronal specific. So what we did, that's the general principle. We have using ExoQuick, which is a commercially available, not expensive. Not expensive. I'm not in Harvard, okay? So when I say not expensive, it means it's really not expensive. Because I have colleagues working in Harvard saying, this is not expensive, 200,000 euros, nothing only. I say, yes, we buy 3 of them every week. So it is really not expensive. So what we do, we basically immunoprecipitate in a way and separate the neuronal, for example, this based on the protein they express in their outer membranes. This we can do it also with microglia, astrocytes, and different type of, if you are targeting myocardic cells or any other cells, you can do it from others too. What I want to show you in this slide is that just two publications showing that just based on the brain exosomes collected in the peripheral blood, we can distinguish patients that are belonging to the Alzheimer's disease or they're healthy, based on the amount of tau and amyloid beta they have in the cargo of their exosomes. And that's another, let's say, longitudinal study where they took blood every 10 years, so in a period of 40 years, and based on again on tau and other cargo of the exosomes, they were able to predict the cognitive decline of these people. So suggesting that they don't only have diagnostic help, but also they can have prognostic. In other words, what I was telling you, that now I have started Alzheimer's disease, and probably I have no symptom, but probably it has started because I can read through my blood exosomes, brain exosomes in the blood. And indeed, for people working in Alzheimer's, for example, last July, blood is included in the diagnostic tools for Alzheimer's. And I'm sure in the very next year or in 2 years' time, we're going to have a panel of proteins and microRNAs in our brain exosomes in the peripheral blood that will help diagnosis. Not only for Alzheimer's disease. This is exosomes. I'm giving you an example because I work on that, but it is a platform that can be used for any kind of disease. So we are about time, almost there. Okay, so in this review, I'm trying to show you the main two things that we have that happened in the Alzheimer's disease brain. We can also find, sorry, in the exosomes of the brain collected in the peripheral blood. So what is the first thing that I told you? First thing that I told you it happens in the Alzheimer's disease brain. You answer the question when I asked you. Atrophy, huge atrophy. So atrophy means I lose dendrites, I lose cytoskeletal proteins, I lose spines, synaptic proteins. What do I have inside the cargo of the brain exosomes in the periphery in Alzheimer's disease patients as we progress? Reduce synaptic proteins. What else we have? Main molecular changes in the Alzheimer's disease brain. Loud. Ella, you can. Hyperphosphorylation of tau, accumulation, and amyloid beta. What do I have inside the cargo of amyloid beta, of exosomes, brain exosomes in the periphery? Exactly that. Amyloid beta molecules like this, phosphotau, total tau, and others. Exactly. In other words, the cargo of the brain exosomes collected in the peripheral blood can mirror, can reflect the pathological changes happening in the brain. And I will move now a bit to the stress, as this is a very important parameter of risk, a risk factor for the precipitation of Alzheimer's disease. I told you from the beginning. I don't want to stay into many details. I just want to show you, another thing. I want to show you that previous work from us and others have shown that if you expose animals and humans to long period of chronic stress, we have an activation of amyloid beta pathway, and this leads to synaptic degeneration, as well as we also have the different phosphorylation, accumulation, different parameters of tau pathology that are activated by chronic stress. So, this accumulation of pathological forms of tau is happening because chronic stress is inhibiting the endolysosomal pathway as well as the autophagy. Both are finishing the lysosome. And as I told you that the endolysosomal pathway is involved in the exosome release, we recently saw that this also affects, that chronic stress also affects the secretion of exosomes from the brain. And this is indeed reflecting, this is analysis, photomic analysis of the exosome from the brain in stressed and controlled animals, and they more or less represent what happens in the brain. Atrophy, problems with degradation and the stuff. I don't want to make a long story short, but so, and finishing the talk. So I tried to say you all this time that complex diseases such as Alzheimer's disease, depression depend on the interaction between genetic and environmental parameters. And we have shown that Alzheimer's disease pathology and stress-related pathology share some same mechanism that leads to synaptic and neuronal atrophy and cognitive and mood impairment. And we use different ways to approach this, including exosomes from mesenchymal stem cells, including exosomes that they can be loaded. And how this can happen, and I'm finishing, this can happen because, for example, by isolation, I can, collect from mesenchymal stem cells different exosomes that I can, after characterization, as I told you with electron microscopy or flow cytometry, after this, I can deliver or load with drugs and deliver back to the brain. For example, we do it with animals, and we put the back exosomes in the peripheral blood, and they cross and they go to the brain. We label them, and we see that they go. What it is also very interesting that there's a lot of effort where researchers are trying to modify the membrane of the exosomes, and they say, you deliver this drug only to microglia cells in the brain. How? By bioengineering the membrane with one specific protein that is found in the surface of microglia or the extracellular space related to microglia. And in that way, we can have, it's not really in very well established, but there is a lot of effort on that direction on bioengineering. And I think I'm done. That's different applications of how you can treat exosomes. I don't have more time, but you see a lot of different applications of exosomes in regenerating or trying to help disease conditions in different organs and tissues. And for that, I want to close with MNS and to invite you to come to Crete, Chania. Greeks are the few Europeans or the only that can say in their language. So Chania, for Tunisian people, it's easy to say. It's in Crete, it's South Greece. It is beginning of June. So there are a lot of different fellowships and travel grants. Actually, it is very cheap. We managed this year to have very, very cheap, like 50 euros registration for 4 days for master and PhD students. Go on the website, check it, and also apply for travel grants. There will be IBRO travel grants, there will be MNS travel grants. So for you to join it. Okay, so it is June from the Mediterranean Neuroscience Society. And I think I'm done. Yes, that's the people of the lab. So the main lab now for the last 3 years, I moved back to Greece, but also the previous lab in medical school in Portugal is also on with Joan. I've been the young group leader. So we are working as one family, what we call 2 teams, 1 family. And thank you very much. That's our collaborators. Thanks again.
Thank you very much, Jonas, for this wonderful conference. Alzheimer's disease is one of the scariest diseases, in my opinion, because then you end up at the end of your life when, if you have, not you, but if someone has it, so not recognizing the people you love, that's something I find really, really scary. I have a question, a little bit philosophical, a little bit, if we can call it like that. So recently I have been into a training about brain health, because we work a lot at to train us on how to keep our brains healthy. So, and one thing that, one sentence that I really loved is that the brain is 200,000 years old. The human brain is 200,000 years old and is not made, it wasn't made or conceived for this amount of information that that it has to treat on a daily basis. The trainer at that time. So he said, do you think that this is something, this is something accurate, first of all? And second, do you think maybe Alzheimer's disease or dementia, is it some kind of an evolutionary process somehow that is happening actually now in order to remove the brains or that are not able to, are not...
Got it.
Yeah. So that's my...
Okay. So I don't think that there is a limit for the brain process. Actually, it is one of the most adaptive and plastic organs that we have in our body and in the whole planet. I don't know about the rest of the planets and universe, but in this planet, brain is the most plastic. And I can tell you a very simple example for people working in Parkinson is well known. For example, in order to have this tremor problem with Parkinson people, you need to lose from one specific area of your brain, at least 70% of these dopaminergic neurons in that area. So up to 60 something, despite the big neuronal loss, the other neurons, the other cells, they modify their actions in order to manage to do the job. This is huge plasticity. So I don't think there is a limit in the way that the brain processes. What can happen is that is overworking. That's why when you're under stress conditions, you forget information that is not prioritized as important. Or there is no more space in the hard disk. That's why you delete it. But that's absolutely normal. That's absolutely normal. There is a big process how the brain, filters information, store it, restore it. Okay, I had a card with this PIN number. This expired and a new card arrived. So I know that my bank 4-digit number is not anymore the old one, but I replace it in the same place with the new number. So there is a lot of endless process for the brain. So I don't agree that Alzheimer is something we see now. Second, it is an age-dependent disorder. And the longer we are living, the higher the percentage to develop age-related disorders. Same for cancer. Now, if our society or way of living had changed and we have increased these factors of the stress, it imbues it. We can speak about the gut-brain interaction of what we eat and how these and nutrition habits and sleeping habits, this can be, precipitation factors, not only for Alzheimer, but for damages of the brain. Now, if you ask me why we are under the same stressor, why you don't develop Alzheimer and I develop Alzheimer, that's the issue to understand, the difference in vulnerability in diseases. And this is why precision medicine, in many years, we will have these answers because we have collect big data in order to compare. So,
thank you very much for this insightful response. So, are there any questions in the room? Yes,
Thank you for your presentation. Regarding the use of exosomes and extracellular vesicle as attractive alternative to cellular therapy, one of the most limitation of their use is related to the lack of standard protocol in preclinical isolation. Do you think that we can in the future, we can standardize the preclinical isolation?
yes and no. I will explain you why. The protocol we are going to, let's say the method we are going to follow in order to isolate exosomes, it may depend on what is the purpose of use and how much you will need. For example, your surgeonist, my ophthalmologist surgeonist, Tunisian friend, I met him yesterday night. So he told me they have a bit different laser machine to cut something sharp. I don't care for side effects because I need to cut and remove the finger that is going out from my eye. Well, it's very, very different with the new Leica machine laser, but they remove one scarf they have here in the edge. They need to be accurate, no bleeding, and very fast. So this example says that it depends on the application. You always lose something. You get a lot of stuff, but less purity. You get less stuff, but more pure. So it's true that it's a mess. So that's why in science, and especially with working with exosomes, even for targeting new stuff or putting drugs, you need to check carefully what are the standards that these people publish using. Did they check how many things they did to check what they get? They say isolate exosomes. Yeah, and I'm the president of the United States. Show me proof. What have you done? Did you do this level, this level, this level analysis? Then I use the protocol. But otherwise, I don't trust it. And I don't say that these scientists are bad. It's that say, as a scientist, I have the right to doubt about myself, doubt about the others. That's how science goes on. Nothing wrong to that, okay? If Alpha told me, oh, John, yes, I like your technique, but I think there's a better one. It's not a personal accusation. There is a better because For her application, she needs a better application, a better technique. Okay?